Phylogenetic Analysis of the ERA-A Gene in Endomicrobium persinimphae from R. flavipes by Serena, Kelsey, Licketh, and Kyle. In our study, we explore how lower termites consume wood through a symbiotic relationship with celluloidic protists. These protists found in the hindguts of termites like Verticulatermes, flavipes, host bacterial symbionts of their own, such as Endomicrobium persinimphae. By analyzing the genes within the endomicrobium, we look at the genes responsible for wood digestion. Our focus is on tracing the evolutionary history of the ERA-A gene within the endomicrobium, revealing Lactococcus as the likely donor. Thus, we gain insight into the mechanisms enabling termites to thrive on wood. The endosymbiotic relationship between termites and protists is estimated to have formed approximately 40 to 70 million years ago, as noted by Stevens et al. Central to this relationship is the ERA-A gene, pivotal in metabolizing arabinose, a key component of wood. In arabinose metabolism, ERA-A operates within the phosphotransferase pathway, crucial for wood metabolism and overall survival of both the endobacterium and the host termite. Our focus bridges the gaps in ERA-A's evolutionary narrative, exploring its origins within the endomicrobium persinimphae. Through various tree-building methods and phylogenetic data comparisons, we aim to pinpoint ERA-A's probable donor.
For our methods, we started with the gene RA from the ASM 2032804V1 genome, which was blasted using megablasts from NCBI. Blast hits with a percentage identity above 75% were then implemented into a distance tree as seen in Figure 1, and their aligned FASTA sequences were downloaded. The FASTA aligned sequence was then uploaded to IQ-Tree with the default option selected to compute for the maximum likelihood phylogeny for RA blast hits, as seen in Figure 2. Support for nodes was assessed from 1,000 ultrafast bootstrap replicates, which involved generating multiple datasets by randomly sampling observations with replacement from the original dataset. Phylogeny of 16S rRNA genes was created for comparison to the R phylogeny because the 16S gene is highly conserved across bacteria. 16S rRNA genes were extracted from each ARA megablast hit genome from GenBank. 16S sequences were aligned using the MAFT web server, and their maximum likelihood phylogeny was estimated using IQ-Tree, with the default option selected. Support for nodes were finally assessed using 1,000 ultrafast bootstrap replicates.
So in order to investigate possible donor candidates for horizontal gene transfer, we inferred 3 phylogenetic trees. First, in Figure 1, we generated a distance tree from the megablast results of the RA gene region from the Endomicrobium persinimphae genome. And the distance tree infers their evolutionary relationships based just on pairwise distances, so it's based just on the similarity of their nucleotide sequences. And in the distance tree, the RA from Endomicrobium persinimphae groups within the clade of the RA gene from Lactococcus. But in order to compare tree building methods, we also generated a maximum likelihood phylogeny in Figure 2, based on those same megablast results for the RA gene. And the maximum likelihood tree building method incorporates more robust statistical analysis, and it optimizes the tree based on the given data set. And in the maximum likelihood tree, we can still see that the RA gene from Endomicrobium still branches within the Lactococcus clade, but instead of being nested within, it is now the outgroup of the clade. And lastly, we generated a phylogeny of a non-protein coding gene that is not from horizontal gene transfer, and we compared that to the RA gene tree. So Figure 3 is a maximum likelihood phylogeny of the 16S rRNA gene from all of the same organisms as the first 2 trees, and we can see that Endomicrobium persinimphae is actually most closely related to Paenibacillus and Lacno-anaerobaculum as opposed to Lactococcus like in the RA gene tree.
Overall, the 3 phylogenies produced in this study show different branching orders depending on the tree building method used and the gene of interest. Both the distance tree and maximum likelihood tree show that Endomicrobium persinimphae's RA is most closely related to the RA genes found in Lactococcus. The exact phylogenetic position of Endomicrobium persinimphae differs in these 2 trees because the distance tree uses neighbor joining, while the maximum likelihood tree calculates the most probable tree topology. Figure 3 alternatively shows that the 16S rRNA gene of Endomicrobium persinimphae is most closely related to those of Paenibacillus and Lacno-anaerobaculum. Since Endomicrobium persinimphae's RA shows close relation to Lactococcus, despite the fact that Endomicrobium persinimphae is more related to Paenibacillus and Lacno-anaerobaculum, we infer here that RA was donated to Endomicrobium persinimphae by Lactococcus. The original study that we're critiquing claimed to reveal the evolutionary traits of Endomicrobium persinimphae, but failed to identify horizontal gene transfer donors. Here we expand on their findings by comparing phylogenies and determining that RA was most likely transferred by Lactococcus, bacteria which are also found in the termite gut environment. This indicates that Lactococcus and Endomicrobium persinimphae may share similar niches within the termite gut environment. Because bacterial RA aids in wood digestion for the termite host, these findings can broaden our understandings of how selective pressures from the host can influence the survival and maintenance of microbiota, which accept genes that contribute to overall host function.